Should put you in the waiting room, Justin. Like timeout. All right, CATLab 12 is the one that's due today. We've gone over CATLab 12, but is there any lingering questions on CATLab 12? I want to spend the majority of the time going over the mitosis POGL, but CATLab 12, any questions on CATLab 12? I don't hear any questions. I know you're not muted. I only have 4 turned in. That's why I'm asking. CATLab 12? You guys in a coma, or you guys just have it all done and don't want to waste your time with CATLab 12? Is your audio working? Can't hear anybody.
I think everyone's just muted.
Okay. Justin, you're okay with CATLab 12?
Yep.
Okay. Michael, CATLab 12 is good? Oh my gosh, what is going on? Morning, Jesse. Okay, CATLab 12 was the first one over the digestive system. So Jesse, I just asked if anybody had any questions about that. It was about yawning, contagious yawning, hiccups. Had to write a paper about hepatitis, I think, on that one. And job of the liver and gallstones. Acid reflux. Did you find anybody find anything about GERD? Anybody find anybody that was diagnosed with GERD?
Daniel Davis.
Oh, that was like 8 years ago. That won't count. That was when the question originally developed, but there's somebody much more recent than that. No? All right. So this is your last chance, CATLab 12. Everyone's going to have it in today, right? Because we went over it, but I'm opening it up for questions. 10 more seconds. Okay. Open up the mitosis POGL. That is due tomorrow. I will share part of my screen. You should be working on it as we're working on it. Please don't do the same thing. Oh. All right, every time I try to share part of my screen, it does this still. I don't know what I'm doing wrong. Hold on. I just want to share the lab, and it's like, sorry. There's something here that I guess I don't understand. Well, whatever. We'll just get by. Your faces will be like part of the screen here. Hey. Oh, and you can't see that at all, can you? Oh my gosh, stop. Okay, Michael, let's start with you since you're the top of my screen anyway. List the 4 phases of mitosis. Can't hear you.
I was muted.
Okay.
Prophase, metaphase, anaphase, and telophase.
All right, prophase, metaphase, anaphase, and telophase. Oh, come on. Okay, Tyson, you're up. Where is mitosis in the cell cycle? Before what and after what? Mitosis is right here.
I put G1 cytokinesis and then after G2.
Okay, so it's before G1 and after G2. Okay, Kylie, what are the three phases of interphase?
G1, synthesis, and G2.
Yes, G1, synthesis, and G2. Okay, Austin, how many cells are present at the beginning of mitosis?
2.
Like at the beginning, though.
Oh, I have no clue.
So right here,
1.
just one. Okay, Drake. Oh no, sorry. How many cells are present at the end?
2.
Okay, Jenna, refer to the chromosomes in the cells in Model 1. How is a single chrome represented in Model 1? A single line or an X?
And an X,
Let's try again.
a single line.
A single line. Yeah, I mean, you see the X's, but a single line, like just like one / line is like a single chromosome. Okay, replicated chromosome, Zaya, is
X.
an X. Okay, how many chromatids, Justin, are in each replicated chromosome?
Two.
Exactly. All right, let's see who's off the screen. All right, Kaylee. Hey, how many replicated chromosomes are in the original cell shown in Model 1 during prophase? When
4.
counting chromosomes, count 1 for a pair of sister chromatids. So how many replicated chromosomes?
4.
Huh?
4.
4. Okay, Clayton. How many single chromosomes are in each of the new cells in telophase?
4.
4. Okay, as a group, since we're a group altogether, grammatically correct sentence that explains what a chromosome is and why it's important. So a chromosome is made up of what? Anybody, I guess, since it's a group one. What is a chromosome made up of?
DNA.
DNA. Okay. Chromosome is made up of DNA and proteins and contains what?
The genetic code.
Yep. All right, Thomas. Use a complete sentence to describe what the new cells in telophase might contain if replication of chromosomes did not occur before cytokinesis?
They might not contain the right amount of chromosomes.
Yeah. Oh. Over there. Okay, Jesse. If the situation in part A occurred, would the new cells be viable? Would they be, living? Would they be able to carry out their function?
No.
Absolutely not. I'm going to say without a complete without complete correct amount of DNA, they couldn't carry out their life processes. All right. Did we get everybody? Anybody not go? Okay, let's go back to the top. Michael, all about you. Okay, the S phase stands for synthesis, which means to make or build something more complex out of simpler parts. Scientists know that during the S phase, DNA is being made in the nucleus of the cell. Why do you think the cell needs to make more DNA at this time in the cell cycle? Why does it have to make the DNA before it has to start dividing?
It has to be there in order for able to divide.
Okay, well, yeah, I mean, it has to make sure that the new cells have a complete set. Okay, Drake. The chromosomes that are shaped like X, made of 2 sister chromatids, have double the amount of DNA than the chromosomes shaped like the 1 or /. During what part of the cell cycle do you think the chromosomes are replicated?
During like the synthesis.
Yeah, during the S phase or synthesis phase. Okay, Kaylee. Refer to model 1. In which phase of mitosis do you see the spindle fibers forming?
Prophase.
Prophase. Justin, at what phase of mitosis do the replicated chromosomes separate? They start pulling apart.
Mitosis?
Yeah, I know mitosis. What phase of mitosis?
Oh,
So look up the first diagram.
phase,
Oh, where'd it go?
anaphase.
Yeah, it is anaphase. I must have deleted my diagram. Okay, Kylie, in which phase do you see the spindle fibers have disappeared?
Telophase.
Telophase. Zaya, what's the purpose of the spindle fibers?
They separate the chromosomes so that
Yeah.
they're on opposite sides.
Okay, Clayton, describe what happens to the nuclear membrane after prophase.
It's kind of pulled apart so that the
Yeah.
spindle fibers can attach to the chromosomes.
Yeah, it kind of like disappears, doesn't it?
It says it disintegrates.
What says it disintegrates?
The gizmo said it disintegrates.
Yeah, disappears, disintegrates. This, I don't know how to spell disintegrates. I should have just stayed with disappears. Disintegrates, there. What? Oh, I don't know what that means. Ugh. Oh my goodness. All right. All right. Tyson, explain why is it necessary the nuclear membrane disintegrates. Oh, it's right there. Sorry. Disintegrates during mitosis.
I put that the nuclear membrane, it needs to like be removed at times.
Yeah, the chromosomes have to move, right? And so the nucleus can't be in the way. Well, we need to remove the barrier. Jenna, at what point during mitosis has the nuclear membrane reformed?
Telophase?
Yeah, at the very end of telophase. Are we keeping up okay? All right. Thomas. Oh no, Austin. What is actually dividing during cytokinesis? Think of the word cytokinesis. It's the division of the what?
Cytoplasm.
Yes. Hey, Thomas, cellular division has two parts. Mitosis is the division of the nucleus. Cytokinesis is the division of the cell into two new cells. Explain why mitosis has to come before cytokinesis in the cell cycle.
Mitosis has to come before cytokinesis because if cytokinesis happened first, then the cell would fill up with too much cytoplasm
Okay.
first.
And there's a limit to how far cells can grow. So we can't. Oh, my goodness. Okay. Cadence, are you here?
Yes.
Okay. So guess it's all your turn. 16. During cytokinesis, the chromosomes unwind and become a pile of very long thread-like DNA, and the cell goes back to looking normal. Why must the cell coil up into chromosome structures before it divides?
Because they have to be on opposite sides of the cell before dividing.
So, okay, so why must the DNA coil up into chromosome structures before it divides? So yeah, if we look at a normal cell, we're not gonna actually see chromosomes. So it has to like, I guess, make itself into what was that? I don't know. Make itself into smaller compacted areas, I guess, so that DNA can replicate better or easier. Sorry. What's wrong with that? No. All right. Fill in the table where each of the phases of nuclear and subdivision is listed in the left-hand column. Write a description of what's happening. So, okay, prophase. Michael, what happens during prophase? We just talked about the chromosomes. The chromosomes do what?
They replicate.
Okay, that's during synthesis, but the chromosomes become starts with the V.
Visible.
Yeah. And I guess the other good thing here is the spindle fibers form. And what happens to the membrane?
Still me.
Yes, sure. So during prophase, the chromosomes become visible, spindle fibers form, and the nuclear membrane starts to, whoops, starts to what?
Start to disappear.
Yeah. Okay, Drake, metaphase, what happens?
Chromosomes start to like line up.
Yeah, chromosomes line up in the middle. And what attaches to them here?
Is it the spindle fibers?
Yeah. Okay, Kaylee, anaphase is you.
The chromosomes are being pulled apart.
Yep. And Justin, telophase.
They start to multiply.
What forms in telophase?
A new nucleus.
Yeah. And cytokinesis. Kylie?
The cytoplasm splits, that makes 2 new daughter cells.
Okay. Now, and we're concentrated on animal cells. In a plant cell, since we have that cell wall, a cell plate forms in a plant cell. Okay. Zaya, in some cells, mitosis occurs without a cytokinesis. What would they look like?
It would be multi-nucleated.
Okay. So they would have more than one nucleus. Right. This happens a lot. We like look at, you know, fungus cells. There's a lot of multinucleated cells in a fungus. And there's some cells in our body that are multinucleated on purpose. And there are some cells in our body that don't have a nucleus, like our red blood cells. All right, Clayton. Importance of mitosis of cells in a skinned knee and during the growth of a plant. Why do we have to have mitosis happening in a skinned knee?
The knee needs to heal and the plant needs to grow, so both gain cells.
Okay. I must be 50% dyslexic. All right. Not like there's anything wrong with that. So, all right, Tyson. Consider the effect of the cell if the sister chromatids did not line up correctly during metaphase. For example, if some lined up side by side instead of single file, how might that affect the cells?
I put like the division of the chromosomes didn't divide like accurately.
Mm-hmm. So if chromosomes didn't divide accurately, you know, some of the daughter cells might have too many chromosomes. I guess some would have too little. Right, lead to some type of genetic disorder. All right. Jenna. Colchicine is a poison that acts to inhibit the development of spindle fibers. Uh-oh. Describe the effects on mitosis in a cell that has been treated with colchicine. Colchicine.
They probably wouldn't get spindle fibers, so they wouldn't be able to divide.
Okay, so chromosomes would not divide correctly, yeah. Or chromosomes wouldn't be pulled apart correctly. What am I? Okay. Austin. Binary fissions, cell division in prokaryotic organisms, bacteria which have no nucleus. In addition, prokaryotic cells typically have only one circular chromosome. Predict how binary fission in prokaryotic cells might be different than mitosis in eukaryotic organisms.
I have no clue.
Huh?
I really do not know.
No. Mitosis is pretty complicated. If we only have one circular strand of DNA, it's not going to be that involved. So maybe not as many steps in binary fission. All they have to do is duplicate the chromosomes and then like just kind of split apart. And don't have to worry about a nucleus forming and reforming. All right. So there should be no excuse for us not having that one all done. I'm going to stop share. Maybe. Okay, I want to talk a little bit about a link that I put on your Google Classroom. Make sure everyone can get to it. So if you look at Google Classroom real quick for me under lab directions. And I know we're backing up, but it's going to be the same thing. There's some things I put on here to make some diagrams easier. If you click on, for example, Lab 9B, diagram of knee, and I know Lab 9B, you're done with it, but whoop, never mind, don't do that one. Okay, do heart coloring vein diagram. If you click on that, hopefully that leads you to a class kick. The class code should already be there, and all you have to do is just type in your first name.
Just our first name?
Yep, just your first name. So you notice did everybody get that open okay?
It's still loading.
Okay. It's going to be a page of a heart diagram. All you have to do is like in the box, just use the words red or blue in the box to indicate if they're oxygen rich or oxygen poor. And the second page is, I think, hopefully a vein diagram to label. And that should be a drag and drop. So on that second page, you can just, drag the words to their correct spot. Now you'll notice are you looking at that, or is that loading okay? Okay, on the second page, you notice that on the left side of the heart, there's a couple boxes that they say like directs dorsally or directs ventrally. So directs dorsally, I mean, you can go ahead and put that correct vein there. That's the vertebral artery, or sorry, vein. That's directing, towards the back. But, and as you do this, it'll save your work. The one that directs ventrally right underneath that would be the internal mammary. So I'm putting those kind of diagrams. I think in the lab direction, I put to do the class kick. If you just do it on class kick, then you don't need to really insert the diagram into the lab because, the class kick, I'll see your progress on it. Speaking of labs, I have also put on, so lab 12 is due today. Lab 13 is due on Thursday. You'll see lab 14, lab 15, and lab 16 are all posted on your Google Classroom, and I think the videos for those are done. And the way I'm looking at this and thinking about the schedule is lab 16 is going to be urogenital, so kidneys, urinary bladder, that kind of stuff. And then lab 17 will be your last lab. Lab 17 will be female and male reproductive. Fortunately, I guess for me, we only have a female cat. The male cats that you may or may not have had are already in the dumpster. So we'll only actually dissect the female cat, but I will ask questions about the male cat's reproductive or, male reproductive system. All right, any questions on lab progress? Okay, so yeah, there's a lot to do. Go ahead and feel free to work ahead. Most of the questions on the last 3 labs, I've put questions about the videos. So you're gonna have to refer to the videos to answer them and, get the measurements of certain organs. Reach out if you have any questions. So last day of school is gonna be the 8th, what they said. So then that next week, if you don't have all your labs done, then we can just spend some time I would prefer you guys just finish on time. That would be great. Okay, Kaylee, what's on your mind? Nothing? All right. Okay, that's all I got. So if you want to, hang on and ask me any, questions or talk to me about any problems you're having, you're free. Otherwise, you can go without any hurt feelings. Drake, got a question?
It won't let me leave.
Oh, good. Just ask me a question then.
How's your day been going?
Good. Jesse, got a question?
I do.
Good.
For lab 8,